Welcome to Biochemistry Basics. Subscribe and join us as we uncover how hemoglobin carries oxygen through your body.
How does hemoglobin pick up oxygen, change shape, and release it where your cells need it most? Let's find out.
Textbooks often depict proteins as rigid, frozen architecture. In reality, these are active machines. They vibrate, flex, and breathe, with amino acid side chains moving on a nanosecond scale. This motion allows for induced fit. When a ligand approaches, the protein's binding site physically adapts its shape to wrap around its target. Myoglobin is the baseline for this process. It is a simple, single-chain protein found in muscle tissue, where it stores oxygen for physical exertion. While it binds oxygen with high affinity, that efficiency makes it lethal as a transport molecule. If blood relied on myoglobin, the protein would saturate in the lungs and never release its cargo to the oxygen-starved tissues. Efficient transport requires an internal mechanism to regulate affinity. The protein must act as a mechanical sensor, triggering a release when the environment changes. Physiological transport happens across two extremes. In the lungs, the partial pressure of oxygen is high, around 13.3 kilopascals. In peripheral tissues, that pressure drops to about 4 kilopascals. These requirements are satisfied by a sigmoid or S-shaped binding curve. This graph maps the partial pressure of oxygen against the percentage of occupied binding sites. This contrasts with the hyperbolic curve of myoglobin, which remains nearly flat across both pressure zones. The S-curve drops sharply in the low-pressure zone, delivering a far greater volume of oxygen where it is needed. A single subunit protein cannot mathematically produce this shape. It requires positive cooperation, a multimeric structure where binding at one site increases the affinity of the others. Hemoglobin achieves this mathematical curve through structural physics, utilizing a mechanical chain reaction that starts with a single atom. This reaction begins in the sub-nanometer core of a single deoxygenated hemoglobin subunit. The subunit contains a prosthetic group called heme. Without oxygen, its organic ring is puckered. The central iron atom is too large to fit, protruding downward. When oxygen binds, it initiates a chemical shift. The iron slips into the ring's center, snapping the heme flat. This shift creates a mechanical lever. The iron atom is tethered to a proximal histidine residue located on the F helix. As the iron moves, it drags the histidine with it, pulling downward on the entire F helix secondary structure. A microscopic coordinate covalent bond has been converted into mechanical kinetic energy that propagates across the protein backbone. The movement of the F helix applies stress to the quaternary structure, specifically at the interfaces where the alpha and beta subunits interlock. Deoxyhemoglobin exists in the T state, clamped by a network of electrostatic salt bridges between unlike subunits. These bridges hold the tetramer under tension, restricting oxygen access. The F helix tug creates physical strain against these stabilizing bridges. As a second oxygen molecule binds, the accumulated mechanical strain overcomes the electrostatic energy of the salt bridges, snapping them open. This is the breaking point. The entire structure shifts as the dimer pairs begin sliding and rotating past each other. The result is the R state, or relaxed state. The central pocket between the beta subunits narrows, and the rotated dimers align into a new high-affinity conformation. This physical rotation is the moment the protein switches states. Chemical affinity is the direct result of structural mechanics. The R state provides a new environment for the remaining empty heme groups in the tetramer. The macroscopic rotation forces the remaining heme groups into a flat, high-affinity conformation before their oxygen molecules have even arrived. While the first oxygen had to overcome the tension of the salt bridges, the third and fourth molecules bind with exponentially less required energy. This mechanical reality is the physical manifestation of positive cooperativity, linking back to the mathematical sigmoid curve. Hemoglobin's efficiency is a mechanical domino effect, where initial binding makes subsequent interactions easier. Hemoglobin also tracks metabolic demand, detecting exactly where it needs to release its cargo. Actively metabolizing tissues produce high levels of carbon dioxide and hydrogen ions, which lowers the localized pH of the blood. The response is the Bohr Effect. As acidity increases, the curve shifts right, releasing more oxygen. Free hydrogen ions bind to histidine residues, stabilizing the T-state salt bridges and mechanically forcing the protein to drop its payload. Hemoglobin acts as a multivariable environmental sensor, mechanically forced to release oxygen to accommodate acidity. We can categorize this complex transition using theoretical models of allostery. The concerted model assumes all subunits exist in the same state, transitioning simultaneously. In the sequential model, subunits transition one by one, influencing neighbors through localized strain. Hemoglobin utilizes a hybrid of both. Sequential strain drives a concerted macroscopic rotation. These mechanistic models allow scientists to predict the behavior of almost any allosteric regulatory enzyme in the body. The atomic engineering's fragility is exposed when one component fails. In sickle cell anemia, a minor amino acid swap causes the entire kinetic system to collapse. A single glutamate residue at position 6 of the beta chain is replaced by valine. This neutral valine creates a hydrophobic sticky patch on the protein surface, exposed only when shifting into the deoxygenated T state. When these tetramers stick together, they polymerize into rigid, insoluble fibers that puncture the red blood cell. This single structural error ruins the thermodynamic balance of the protein, proving that human life relies on flawless atomic-level engineering. We have traced the journey from the movement of a single iron atom to the physical rotation of a massive 64,500 Dalton complex. While myoglobin is a perfect binder, it is hemoglobin's mechanical tension, its intentional structural flaws, that makes oxygen transport possible. The abstract mathematical sigmoid curve is simply the physical result of severed salt bridges and rotated subunits. A protein's biological work is defined by the transitions between its structural states, rather than any single static arrangement. The biology of respiration is an act of mechanical physics, a continuous structural chain reaction that drives human survival.
Now you know how hemoglobin delivers life-giving oxygen. Like this video, subscribe, and join us for the next Biochemistry Basics chapter.